Module 1, Part 3. In the last part, we ended by talking about some very common histological stains that are used to enhance visibility of intracellular structures. In this section, we are going to look at one very powerful variation on cell staining. It is used very often in cell biology and microscopy, and it is fluorescent cell staining and fluorescence microscopy. Fluorescence microscopy relies on fluorescent molecules, sometimes called fluorophores or fluorochromes. Unique to fluorescent molecule is that they absorb light at one wavelength and emit light at a specific longer wavelength. Example is fluorescein, which is very common fluorophore used in fluorescence microscopy. It is excited at 492 nanometer, which is in the blue range of the visible light. And its emission is at 525 nanometer, which is bright green. So let's look at a very simple diagram of fluorescence microscopy, and then we will go into more detail later. This cell is stained with fluorescein, and the light for the stain is applied, which is 492 nanometers. Then all of the stain will fluoresce, emit at 525 nanometer, which is the bright green range. But there is a problem. Not only does that fluorescent stain emit light at 525 nanometers, but many proteins within the cell are autofluorescent. So they too will be excited by the 492 nanometer, and the proteins will emit light at all different types of wavelengths and therefore produce a confusing result. There is a way to solve this problem. Before the light arrives at the detector, it must pass through a filter. The filter blocks all light except the emission wavelength that you are interested in. So in this example, the filter will block all light except the 525 nanometers. The resulting image is completely black background with bright green stained objects, which is the fluorescent stain coming through. So on this slide is a comparison of a light microscope, bright field, and fluorescence microscope. So looking at the light microscope, see that the light comes from the lamp, is focused onto the specimen through the condenser lens, passes through the specimen, goes into the projector lens, then to the detector. For the fluorescence microscope, the light source is usually a laser. Light passes through an excitation filter and only allows the light of the excitation wavelength that you are interested in to pass through. So if you're using fluorescein, then the filter will only allow light of a wavelength of 492 nanometer to pass through. It is then reflected by some mirrors and focused onto the specimen. The fluorescein in the specimen becomes excited and emits light at 525 nanometer, and this light passes through a dichroic mirror which serves as a filter and only emits light at 525 nanometer and then passes through a projector lens onto the detector, which is a computer screen, or in a simple fluorescence microscopy, the eyepiece. So here is an example of immunofluorescence microscopy by direct staining technique. A live cultured bovine pulmonary artery endothelial cell was stained with a green fluorescing dye that is specifically bound to mitochondria, and the red fluorescing dye is specifically incorporated into lysosome, showing the localization of the two organelles in the cell. There are many cell organelle and component-specific dyes available. A very powerful technique indeed, and a drastic improvement from the conventional light microscopy. Please keep in mind, when we are observing these images or taking pictures of these images, we can see only one color at a time. We can see the mitochondria as green and the red lysosome at the same time. So the picture is first taken only with the green mitochondria. Then on the same cell, by changing the filter of the emission wavelength, we can take the picture of the red lysosome. then we actually merge those two images by using an electronic merge through the computer. So you visualize only just one color at a time, which the emission filter will allow to pass through. There are some very interesting and powerful variations on fluorescence microscopy. I want to talk about three of them. The first variation is the powerful use of fluorescence of ion-sensitive fluorescent dyes. These dyes fluoresce in different wavelengths depending on the concentration of different ions. So an example of an ion-sensitive fluorescent dye is Fura-2. It is sensitive to the calcium concentration in its immediate surroundings. So when calcium is low, the dye fluoresces in the blue range. When the calcium concentration is high, the dye is yellow and sometimes it is orange or red. This is an example of a migrating white blood cell stained with Fura-2. The cell is motile and moving upward, and the calcium concentration is low as indicated by the blue fluorescence. When the cell gets a signal to change the direction and move to the right, can see that the calcium concentration has gone up and indicated by this yellow fluorescence. So the release of calcium into the cytoplasm plays a role in directing motile cells, which we will talk about later when we discuss the cytoskeleton and signal transduction and how calcium does that. For now, I hope you can appreciate how ion-sensitive fluorescent dyes can provide a lot of information about the roles that ion plays in cellular functions. So the second variation is immunofluorescence microscopy. This type relies on the fact that you have an antibody that binds a specific protein of interest. So what immunofluorescence microscopy can achieve is that you can localize individual proteins within the cell and is a major improvement over standard light microscopy. So to identify certain proteins in the cell, you need a specific antibody that will bind only to those proteins. So you say you find a subcellular location of a protein and it is called protein X. The first step is to generate an antibody that will bind specifically to protein X. These are usually done in rabbits. You inject the protein into the rabbit, which is foreign in the rabbit or antigenic. Wait 8 to 10 weeks and check the serum, and it should contain antibodies to protein X, antibody that binds specifically to protein X. The antibody may need to be purified further for better specificity. However, the procedure is fairly routine. You don't have to use just rabbits. You can use mice, rats, chickens, goats, and even llamas. There are commercially available antibodies from llamas, which are very good for producing antibodies, and they generate quite a lot because they are bigger animals. Just for your convenience, the inset shows a typical antibody. The FAB region is variable, and the tip of the FAB region in red binds to the epitopes of the antigens and potentially could bind to any antigen epitope having billions of variations. On the other hand, the FC portion is the FC region is constant and among all different isotypes of antibody. The most common isotype of an antibody is the protein immunoglobulin G, or commonly known as IgG. So think about that. This is an IgG from a rabbit, and all rabbit IgG will have the identical FC region. So you have the antibody and you are ready to carry out immunofluorescence microscopy. The cell of interest and these red dots are representing protein X. So the first step in immunofluorescence microscopy is to fix or permeabilize the cells. Remember, the cells are in a dynamic state, so to get really good and accurate images, you need to freeze them in time. If you are using individual cells, you can move to the next step, but if using tissues, you will need to section the specimen. Then add antibody that binds specifically to the protein of interest or protein X. And this is called the primary antibody. Since the specimen is permeabilized, the antibody will pass through the plasma membrane and interact directly and bind with protein X. The next step is to add a secondary antibody. It has two unique features. Number one, that the secondary antibody will bind specifically to the Fc portion of the primary antibody. The Fc portion is the part of the primary antibody not binding to protein X. You can purchase secondary antibodies that are specific for the Fc region of your antibody. The second unique feature of the secondary antibody is that it is covalently bound to a fluorophores such as a fluorescein. So you add the secondary antibody and it binds to the primary antibody, which is binding to protein X, and it will light up those proteins in the cells. Then you can visualize the cells or the tissue specimen with fluorescence microscopy. Question arises, why use a secondary antibody? There are two reasons. First, when you conjugate a fluorophore, such as a fluorescent, to an antibody, you destroy it by denaturing a large portion of the antibody. There are huge losses. So if you take your primary antibody that took 8 weeks or so to produce in a rabbit and was an expensive endeavor, you add the fluorophore directly to it, you will destroy around half of the primary antibody. It is possible to do fluorescence microscopy that way, but there are huge losses of primary antibody. The second reason for using a secondary antibody is signal amplification. Usually, more than one secondary antibody will bind the primary antibody, and that will enhance the signal. In addition, the secondary antibodies are commercially available: fluorescent goat, mouse, rabbit secondary antibodies. So regardless of the animal you produced your primary antibody in, you can find a fluorescent secondary antibody that you can buy, which is relatively inexpensive. So secondary antibodies save cost and amplify the signal. So here are some examples of immunofluorescence microscopy. A specific protein can be localized in fixed tissue sections by indirect immunofluorescence microscopy. Indirect immunofluorescence microscopy shows this yellow GLUT2, a glucose transporter protein, in the basal, right here, and the lateral surface of the intestinal cell, but they're absent from the brush border of closely packed microvilli on the apical surface of the cells, which is facing this intestinal lumen. And this is an autofluorescence, reddish autofluorescence of the background to give it a nice contrast. Another very powerful variation is the double label fluorescence microscopy can visualize the relative distribution of two proteins. In the double label fluorescence microscopy, each protein is labeled with a different fluorochrome to compare localizations. A fixed and permeabilized culture cell incubated with rhodamine labeled phalloidin, which is red, specifically binds to filamentous actin, and a rabbit antibody reactive with tubulin, the major component of microtubules, followed by a fluorescent labeled secondary goat anti-rabbit antibody in green that labels the primary tubulin antibody. You can do multiple color staining. This type of microscopy can visualize the relative distributions and abundance of two or more proteins. The top left panel shows the fluorescent stained tubulin, which forms the microtubules in the cell. The top right is a rhodamine stained actin, which forms the microfilament of the cytoskeleton. The lower panel shows the electronically merged images Please be aware you or the camera can see two or more fluorescent proteins at the same time, as you are using a filter to allow only one emission wavelength to pass. Here is either green or red. When you see the beautiful dual, triple, or quadruple color images, are all formed by electronically merging them. This is an important concept for your critical thinking questions. In the right, these are cultured nerve cell stained for a specific cytokine that is found in the cytoplasm, stained with a red secondary antibody. The nuclei were stained with Hoechst, a blue nuclear stain, and the cytoskeletal components were stained with a green fluorescent secondary antibody. Here you have a 3-color staining. Some fluorescence microscopes are capable of even 5-color stains. So to do this, you need to use a recombinant DNA technology. Basically, you take the gene of your protein of interest, and using recombinant DNA technology, you fuse an epitope, which is a short peptide sequence like a flag, MYC, HA, hemagglutinin, etc. And there are plenty others. In many cases, the protein function is actually not altered by this tag and it behaves naturally. In a cell, this protein can be detected by a fluorophore attached antibody. Why this is an advantage? The advantage is, from a commercial point of view, you do not have to produce the primary antibody against the protein. Here it is the protein X, which is a substantial time and cost saving. Then anti-epitope like anti-flag, anti-MYC, anti-HA antibodies are readily commercially available, and as it is widely used, the cost is extremely affordable. The last indirect immunofluorescence technique that I have talked about is with fixed cells. And fixed cells are dead as a result of fixation, which is required to permeabilize them so that large antibodies can get into the cell. At some point, you may want to follow a protein in a live cell in real time. So there is a variation on fluorescence microscopy that looks at live cell, and you can track proteins in real time. This type of microscopy relies on a protein made by a jellyfish. They make a protein called green fluorescent protein, or GFP. It fluoresces in the green range. The excitation and emission wavelength ranges are very similar to the fluorescent, which we discussed earlier. Excitation of 492 nanometer and emission at 525 nanometer. So here is a biochemical ribbon diagram. The reason it fluoresces is due to a collection of these aromatic amino acids that form a ring in the center of the protein, and electrons circulate among these aromatic rings and give the protein its fluorescent properties. Many different colors of fluorescent proteins are now available. There are variants on the GFP and can be used for tracking of proteins. One is eGFP, which is enhanced green fluorescence protein. It is a GFP that has a point mutation. One of the amino acid has changed and makes the fluorescence even brighter than the standard GFP. There is also CFP, or cyan fluorescence protein, that is in the blue range, YFP, yellow fluorescence protein, RFP, red fluorescence protein, and all have enhanced versions. Some are naturally occurring proteins from the jellyfish, but some are man-made proteins. Proteins can be labeled and visualized in living cell over time by using recombinant DNA technology to make fusion proteins with naturally fluorescing proteins, such as green fluorescence proteins. Tubes show these emission colors and the names of many different fluorescent proteins. Here is an agar dish is illuminated to show the growing bacteria expressing several different color fluorescent proteins. So you can use these fluorescence proteins to track proteins of interest in real time in live cells. To accomplish this, you need to make a chimeric protein in which GFP is attached to the protein of interest at either C or N terminus of your protein of interest. So to do this, you need to use a recombinant DNA technology. Basically, you take the gene of your protein of interest and using recombinant DNA technology, you attach the gene that encodes, say for example here GFP, to the gene that encodes the protein of interest, protein X. The GFP will fluoresce when excitation wavelength is applied at 492 nanometer and should be able to track the fluorescence of this protein in live cells. In this example, Dictyostelium discodium non-pathogenic amoeba expresses a GFP RAB8. RAB8 is a protein that regulates vesicle trafficking, which we will discuss more later in the course. In the left, you can see this is the fluorescence image. On the right, this is a phase contrast image, and that is a drastic improvement. Another example of how to track live cells. Again, these are Dictyostelium discodium cells expressing GFP-tagged version of actin-binding domain of actin-binding protein, or ABD, and will bind all the actin in the cells. So you can track actin at real time. These 4 panels are time-lapse field microscopy. On the left are the fluorescent images, and on the right is the corresponding bright field images. Each panel is a frame in a movie using video microscopy. In the fluorescence image, the actin is lit up and generally around the periphery of the cell. The main interest was the role of actin in endocytosis, which is a process by which cells take up particles from extracellular space and do it by forming vesicles from the plasma membranes and pinch the vesicle off so they end up in the cytoplasm. So this is a cell undergoing active endocytosis. Frame 1, the red arrow, see the cell is forming an endosome and is brightly fluorescing, indicating presence of the actin. Frame 2, you can see the progression and the formation of the vesicle as the process continues with the invagination of the plasma membrane. Frame 3, you can see that the vesicle has pinched off within the cytoplasm and surrounded by actin. And in the frame 4, the vesicle is still there, but the actin is no longer there. So the experiment shows that actin is very important early on in the formation of endosomes. After the vesicle pinches off, the actin is gone, indicating its function is early in the endosome formation. So you can easily appreciate how powerful GFP tagging proteins and trafficking GFP proteins in live cells. So you can insert the modified GFP into developing organisms at the right time for them to be expressed in virtually every cell. Examples of zebrafish, which can be purchased in some Walmart stores, also in plants such as tobacco plant, and mice. You can see that one mouse is not transgenic or not green for comparison, and these are all developed by these fluorescence proteins. This concludes part 3 of module 1.